As a public company, let me just draw your attention to our forward-looking statements, which you can find on organovo.com on the IR page. I don't think I'm pressing hard enough. Today I want to contain my comments to rare diseases. I believe that's the session that we're focused on this afternoon. And Organovo has a unique, I believe, and very broad approach to treating a range of rare diseases, which I'll discuss. At the heart of our strategy is our 3D bioprinting platform that consists of our ability to isolate and characterize cells through our Samsara subsidiary, combine those cells through our 3D printing platform in a configuration and dimensions and constructs that can thrive and live in vitro and ultimately in an in vivo environment. And I'll describe how we've validated that and where we're headed with these capabilities. The backdrop of what we would like to address in human clinical trials and ultimately therapeutically are a range of inborn error genetic mutation diseases that play out in the liver, typically resulting in a single missing protein or enzyme of critical function to the body. A number of these diseases I've listed here, including alpha-1 antitrypsin deficiency, type 1 tyrosinemia, and OTC deficiency. All of these diseases lead to a progressive and dire outcome in most of the patients afflicted with the diseases, and typically orthotopic liver transplant is really the only solution to patients suffering from these diseases. In the case of alpha-1 antitrypsin, typically in adolescence or early adulthood, and in the cases of type 1 tyrosinemia and OTC deficiency, sometimes as soon as weeks or months after birth. Our strategy is to develop 3D bioprinted functional liver tissue patches that we can implant adjacent to the liver to deliver a broad range of function into these patients. Our hope is to demonstrate that our patches provide both supportive liver function, which is important in patients as they progress end-stage liver disease, for instance, are on a transplant list awaiting transplant, and over time, perhaps to demonstrate that we can also replace the missing synthetic function in these patients, in which case we can either provide meaningfully a bridge to transplant, and a bridge of a few weeks to several months could make the difference between life or death for patients awaiting a shortage of transplantable livers, or perhaps obviate the need for transplant. Obviously, if we can work upstream from that to patients before they progress to end-stage liver disease, all the better. I'm going to have to learn to punch like one sentence before I finish each slide. In terms of opportunity, what I've depicted here are the medical costs associated with treating these diseases that we've modeled out through an external agency. And in the case of alpha-1 antitrypsin disease, which is clearly a rare disease, the cost of palliative care for a year of treatment in these patients approaches $250,000 per patient, represents a medical cost of upwards of $1 billion, and therefore addressable market to those of us that can help to address this disease. If you add in the full range of genetic mutation diseases I've outlined affecting the liver and include acute on chronic liver disease, which also is an often fatal outcome of patients awaiting transplant, you're looking at an addressable market of over $4 billion. In terms of viewing our pipeline, somewhat uniquely, you need to start with the fact that we are creating healthy, functioning liver tissue. That is our product. And profiling this healthy, functioning liver tissue is the first step, therefore, in progressing our pipeline, demonstrating that it can be placed into in vivo systems that will engraft, that it'll thrive, and that it has durability in a living system. And then to take these same patches and apply them to various genetically modified animals, modeling the various diseases of interest to see if we can affect disease outcome. And that's exactly what we've been doing over the last couple of years at Organovo. Our most advanced program is in alpha-1 antitrypsin deficiency, for which we recently received orphan drug designation from the FDA, and we'll be progressing towards an IND filing in the year 2020, which is, in our view of the world, not just around the corner. We've also demonstrated compelling animal data in type 1 tyrosinemia, where we actually show a differential between control animals and the treated animals in terms of survival in that model, which is quite exciting. And there are a number of other areas one would contemplate treating with this strategy, and we certainly will be exploring those as we go forward in the next couple of years. May have a battery issue here. Or could just be my tactile strength is waning after shaking so many hands today. I won't present too much preclinical data, but again, at the heart of our premise is this audacious thought that we can create from a cell-by-cell level a 3D tissue, implant it into a living animal, and expect that animal to accept the tissue rather than reject it, and actually incorporate it into their biological systems. And in the upper histology chart there, what I'm showing is the region of our implanted patch adjacent to the host liver. The yellow arrows actually depict the vascularization that has now connected our tissue to the host animal. This is happening not through our design, but just through the interface of our tissues with the host liver. Moreover, we're able to study circulating human proteins in the animal model, which can only be coming from our functioning tissue. And the proteins of interest, including OTC and A1AT, are present in those animal models. Therefore, and quite encouraged, we went on to test these patches in disease models. And we've been able to see impact on the disease as well as functionality, which has encouraged us and led us to begin the interaction with the FDA on an IND track program. One unexpected and quite exciting finding, and this is from our PIZ animals, which demonstrate or create a model of alpha-1 antitrypsin disease, at least in the liver. The hallmark of this disease are misfolded proteins that accumulate in the hepatocytes, ultimately clogging the function of the hepatocytes and leading to cell death. On the top panel, in the sham animals, you see over the course of 125 days, progression of diseased, clogged, or toxic globule-containing hepatocytes. By day 125, the dark purple stained cells have almost completely overrun the liver. In our treated animals, and these are histology slides taken from the region of the liver immediately adjacent to our patch, quite a different situation has ensued. We see a clearing of the nodules or prophylactic preference for healthy hepatocyte formation in the region of the liver adjacent to our patch. We're not sure what's going on yet, but we believe if we can demonstrate this in further models that there may be a prophylactic or possibly even a curative effect that one might explore in these animals. And this is over and above the fact that our patches will be delivering synthetic function and boosted liver function in a highly diseased liver state. So as I mentioned, we are on a track to FDA filing, ultimately to begin clinical testing in humans with the goal of submitting our IND in calendar 2020. We've had our first interaction with the FDA, the pre-IND meeting, and those of you from other sessions have been hearing about the unmet approaches and other strategies the FDA is pursuing to interact with companies in the regenerative medicine space. The pre-IND meeting is an example of that, which I believe they will now convert to what they're calling the interact process. And it's a great interaction that gives us a very early indication of how the FDA is going to view strategies like this and how we can tailor our IND, CMC activities, modeling, and the like to get through the FDA process in such a novel field. We would expect to have our formal pre-IND meeting with the FDA next year, and we also expect to nominate a second IND track program with a corresponding orphan drug designation also in 2019. I've focused my presentation on the liver, and indeed, Organovo, over the last couple of years, has focused most of its research on 3D bioprinting liver tissues and applications therein. We have quite a bit of proof of concept data in other solid organs, and importantly, we've also, through tech transfer agreements, placed our platforms at a number of leading research institutes to study other solid organ solutions that we hope can lead to further IND track programs utilizing bioengineered tissue, including tissues embedding stem cell and other strategies towards treating a broad range of diseases across some of the organ systems I've cited here. Finally, as a biotech company, we're cognizant of the fact that we're a capital-intensive undertaking. And like many of the companies here at in the regenerative medicine space, the pioneering work we've done on creating our platforms creates an opportunity to partner, generate revenues, and provide or fuel collaborations with other leaders in the liver space. And we have capitalized on this through providing our highly curated and valuable cells from our Samsara subsidiary to researchers in the biotech and pharmaceutical industry. We also license our platforms in a selective manner. And finally, we've created a range of custom applications in our tissues where we are able to keep our tissues alive for up to 4 weeks in an in vitro setting, introduce disease states such as NASH or liver fibrosis into those tissues, and then treat them with promising new drugs. And this has led to funding that helps support our overall research mission. So in conclusion, again, I'd like to thank ARM for bringing us all together today. What I see are 3 major trends all gathered in this meeting. Those that are developing cell-based strategies, genetically modified cell or DNA-based strategies, and tissue-based strategies. But if you think about it, they all mesh together. We all have to kind of work together to progress this industry forward, and we're just delighted to be part of it at Organovo. Thank you very much.